Welcome to the first podcast of Sign Action, a channel where I bring my research to the public as it happens. In this episode, I will give you guys a brief history of how I got into research and why. It all started on the island of Prince Edward Island on the east coast of Canada. And as you see on the photo here, lobster fishery is a big thing on this island. It's also, I guess, then not that surprising that my research was not that far removed from marine biology, even though as I studied biology as an undergrad, plants were my thing. So I really was interested more in plants than in animals or marine animals. However, I started with sort of a combination of marine biology and plant ecology. How it all began was I was just walking up the stairs at the university, and my marine biology professor was walking down the other way, and he said, "Andre, do you have, by any chance, job set up for the summer?" So it's really common for teenagers and undergrads especially to work the whole summer and earn money to pay for their tuition. And I said, "No, I don't have a job at the moment." To which he replied, "Well, you should apply for this fund that exists that allows undergraduate students who have never had research experience to sort of get their feet wet and basically just write a proposal of any research that you would be interested in with the help of any professor from the university to try your hand at research for the whole summer." So I said, okay, fine. I knew that he was a marine biology professor, obviously, but that was not really my thing. So I said, well, I really like plants. Is there anything we can do with plants? And he said, well, come to think of it, there's this eelgrass, which is a really, really important plant for the island and also for other marine systems elsewhere because it provides a very important refuge habitat for many fish and lobster species, which are important for the fishery on the island. So I said, okay, yeah, eelgrass sounds good. And I read more about it, and I found out that these invasive green crabs, which were invading the island, that they potentially were having a detrimental impact on this grass. But it was not clear the mechanism through which they were doing this. So I thought, all right. So I wrote a proposal, and it was successful on trying to establish exactly what the mechanism of this damage was through these green crabs on this eelgrass. And so what I did was, for 2 summers, I basically went to the coast and collected eelgrass plants, and then I planted them into aquariums, as you see on the photo here, and let them grow there. And then I also trapped these invasive green crabs on the beach and brought them in and put them in some of these tanks and videotaped basically what they do sort of during the day and during the night. So I kept a watch on them, and I was able to document how they actually dig through the sand and eat parts of the eelgrass. So they seem to sort of peel these stems or tillers of eelgrass as though they were peeling bananas, so to speak, and to get at the flashy, sugary base portions of the plants. So I was able to show that and write reports on it, sort of describing the mechanism that was responsible for the damage that these green crabs were having on the eelgrass. And that was my experience before even completing my bachelor's in biology. My bachelor's in biology was also related to this eelgrass. Here, I basically, with the same professor, we came out with a project to set up traps at different locations of the island to test what the correlation was between the number and abundance of these invasive green crabs and the quality of eelgrass which was growing in the area. So I would take a truck, drive out to different beaches on the island. Was Not a bad job, I would say. Go for a swim, then set up my traps, and then a couple of weeks later, I would drive again to the same location, take the trap out, which had some fish bait in it, and then I would take all the crabs, bring them back to the lab, measure them, count them, and then I would also do surveys of the eelgrass abundance and health in the same location where I set out my traps. And in the end, I had data on how many crabs at which locations were present, and then also what the quality of the eelgrass was at all locations. And then I was able to show that it was indeed so that the more green crabs were found at a particular location, the worse was the health status of the eelgrass. So together with my undergraduate studies, where I was able to show that green crabs indeed could use this plant as a food source, through these trappings, I was able to then also show that this correlation existed between the health status of this important plant, which provided refuge and protection for many fish species which were important for the fisheries of the island, and that these invasive green crabs were having a negative impact on them through direct feeding behavior. So that was the most novel part of my undergraduate research. So this experience got me really interested in. I thought it was really, really interesting that you could sort of find causal relationships between different environmental factors, which could explain the main cause of decline in an important species in an area. So then I sort of just kind of asked around, okay, what's the next logical step to sort of continue doing this stuff? Maybe that could later lead to some kind of a job. So the logical next step was to find a master's project. And for that, through email, through the internet, through telephone phone calls, I found a professor who was willing to take me on as a master's student. And this was in London, Ontario, in Canada as well. So then I moved there and did my master's there. My research there stayed in the grass vicinity. However, this time nothing to do with marine grass. This time it was just different species of grass which grew in fields around London, Ontario. And here what I researched was how warmer winters could potentially impact these grass species in the area. So the big thing here was that with warmer winters, the area was getting less and less snow, which led to soil freezing and thawing more regularly than during the past when winters were consistently cold. And so this led to potential negative impacts on the grass, where through this increased frequency of freezing and thawing, grass could potentially grow worse in the summer. And one potential mechanism that was hypothesized at the time was that during the thawing and sudden warm spells, the grass was sort of waking up, right, from its winter sleep. And then when the frost returned, then it was caught off guard and its roots were damaged due to these sudden frosts. And this led to a decreased growth. So I quantified exactly during which times and at what intensity was freezing damaging to these grass species which were growing in the area. So as you see in this clip here where I show my methods, sort of how I worked, so where I went to the field side and where I collected clumps of grass, and then I separated them into individual grass tillers, as you see here, and then I planted them sort of in these mesocosms to reflect as best as possible their natural conditions in the field, and then I froze them in these tubes. After freezing them, I washed them again as best as I could, as you see here, And after washing them, having been frozen to different temperatures, I then put them in these glass jars that had nitrogen solution in them. And so I was then able to see, after exposing them to this nitrogen solution, how much nitrogen, right, a nutrient which is very important for growth for plants, how much of this nitrogen in this solution was actually able to be taken up by these plants, by the ones which had been frozen to different temperatures and by the ones which had not been frozen, or the ones that had been frozen to a lower minimum temperature. After exposing the plants to this nitrogen solution, I dried them and sent the samples for analysis, and a company was able then to tell me how much nitrogen was present in different plants. In the end, I was able to show then which temperatures during which time were more detrimental to the growth of these different grass species. So in conclusion, my master's work revolved around looking at how warmer winters could potentially be damaging to different grass species of the area depending on when frost took place during the non-growing season. Following my master's, I was continuing to enjoy my life as doing research in plant ecology. So, again, asked around what's the next logical step, and the next logical step was to do a PhD. So, again, I emailed around through some phone calls, sent my resume with the research that I had done prior to this point related to plant ecology to different profs around the globe, really, because I was not tied down to any particular location. I could go anywhere, really. So for me, location was not as important as sort of the topic. So this brought me to Germany. Here I did my PhD. I continued researching how plants respond to different environmental stressors. Here I focused on climate change impacts on plant growth. And my main question in my PhD was how different plant species, as well as different populations of the same plant species, respond to climatic driven stresses such as drought and frost. So frost stress was this involved warming plants up during the winter. So the project involved growing different grass, forb, and herb species, and then exposing them to different stress factors. And so the stress factors involved either frost after different durations of experimental warming to simulate sudden warm spells in the winter, and also drought stresses. Having imposed these different stresses, my main question was whether different species growing in the same location where I was doing my research, so in the south of Germany, whether different species responded more differently to these stresses, or whether populations of a single species collected from different locations along the distributional range of a species, whether they responded differently to these stress factors. So the reason behind this was to give a sort of a measure to the maximum variation that exists within one species in terms of its responses to a particular stress. And this is important in the future for predicting how species will manage or cope with climatic warming, because if different populations of the same species respond very similarly to a particular stress, well, then it's a lot easier to project or predict its future response. However, if the response of a species is highly dependent on the location from which it comes, which represents then the local adaptation that is present within this species. That makes prediction then more difficult and means that experiments are required to be conducted for each of these different populations, which responds differently. In the end, I was able to find that this within species variation to stress is as high as among species variation. So what it means is that if you had some drought or frost tolerant species growing within the same location, the difference between the frost tolerant species and the non-frost tolerant species was as high as the difference within one species, but comparing its populations from, say, a location in southern Europe and middle of Europe. So I was able to show that it's very difficult to speak of a particular species response to climate change without having tested all of its distinct populations and how they respond to different stress factors linked to climatic warming. Because a species is so variable in its response to warming, depending on what population you're looking at, and it's almost then as important to know what population you're working with as it is to know what species you are working with and how that species will respond to climate change. So that was my PhD work. And then, having finished my PhD, the next step was kind of taken out of my hand, you could say. So similar to how my marine biology professor saw me on the stairs one day and said, "What are you doing this summer?" Similar situation here. As I was coming to the end of my PhD, my supervisor posed this question saying, "I guess you're now without work when you're done with this PhD." And I said, "Yep, that's right. I don't have anything planned." So he said, "Well, I'm moving to a different city and starting a different job there. So if you would like to continue research, you could apply for a postdoctoral position at the same university where I'm moving, and maybe we can continue our research there together." So I did that, and that worked out. So that's where I am now. Now I'm in the north of Germany, and I'm still working with plants. I'm still working with assessing how different environmental stress factors impact plant growth and plant responses to climate change. And my main focus now is on plant phenology. As you probably have guessed, if you have seen the previous video on leaf fall in the first episode of Sign Action. So mainly what I do now is I grow trees in pots, 1 to 4 year old trees, and having grown them in pots, I expose them during the non-growing season to really weird summer-like conditions inside growth chambers or growth rooms to try to get them to open their leaves and start growing. And obviously I can't do that with grown trees, so what I do there is I cut their twigs, as you see on this bottom photo here, and I also put these twigs in water, and I also expose them to these summer-like conditions in growth chambers to see how much heat they need to start growing, to open their buds and for their leaves. And doing these strange things helped me explain why different species start growing at different times in the spring, and how the growing season of different growing tree species may change in the future. So in case that interests you, sort of why I do these weird summer simulations for these tree seedlings and twigs, you can join me on the next podcast where I delve deeper into bud dormancy, which is the process that I currently investigate in these climate chambers. So see you next time on the next podcast of Sign Action.